Carl, in a couple of sentences, what is CRISPR, and why are scientists so excited about it?
This is like totally unfair because I wrote thousands of words on this subject not that long ago, but I'll try to restrict it. So CRISPR stands for something that I can't even remember right now. The acronym is ugly and not worth knowing, but basically what it is gene editing that's cheap and powerful. People have been manipulating genes for 40, 50 years directly, but it's been very crude. Sometimes, you can trying to move a gene from one species to another. CRISPR came out of, actually it was discovered in bacteria. It was basically a way for bacteria to chop up DNA as kind of an immune system against viruses. And then people harnessed it to basically chop out a very specific piece of DNA and replace it with something else. And then you can do very fine editing with it. And it's really only the past couple of years that it's come about, and it's cheap and it's easy. And like I said, and that's its promise and kind of I guess its danger. So that's.
Is it a kind of technology that you can do in your kitchen?
Is the kind of technology you can do in your kitchen. I've seen some Kickstarter campaigns for little CRISPR kits, but if I understand it correctly, that's pretty basic stuff. It's like, hey, use CRISPR to put this gene into some E. coli and it will glow. You're not going to destroy the world with glowing E. coli.
Right. I mean, when recombinant DNA first came along in the mid-1970s, you could only use it in restricted environments, P1, P2, P3, P4 facilities, where P4 was the kind of facility in which they do research on chemical and biological warfare, Fort Detrick, Maryland. And nowadays, most recombinant DNA is in high school labs.
Yeah, there's these contests, iGEM, where the kids get together and they try to just use recombinant DNA to do something cool, like make E. coli smell like spearmint or bananas or something like that. And yeah, it's a science fair thing now. CRISPR isn't quite there yet, but I don't see why it wouldn't be. It's interesting actually talking to scientists about it because they'll say just in their work, the scientists are not trying to change life as we know it, but they just want to study what some gene does, and so they'll put they want to create a mouse with a slightly changed gene just to see what happens, and it could take a year to engineer a mouse and to actually see if that mouse line was really going to work and you could use it. And now it's just, you almost sort of virtually kind of mail away for it. There's this whole step in science that has just suddenly shrunk down because of CRISPR. It's amazing.
Mm-hmm. Right. It's a very powerful technology and probably unprecedented, I think, in the ability it gives human beings to intervene in the gene, not only their own genome, but the genome of every living species on the planet.
Yeah, that's the other thing, is that some of these gene editing techniques, they would work really well in some species or some group of species, you can use this in bacteria that have some way of trading genes with each other, and that's it. Or you can use it in mammals, but in CRISPR, it's nobody has found anything where it doesn't work.
But just to give you one final point here, and then we'll move on. It used to be that you could isolate genes, and then in order to insert them in another organism's genome, you had to insert the gene into a viral vector, which would then carry the gene into the genome. But you had no control over where it would go, or whether it would function, that is, express itself, or mess up the genome.
Yep.
What's really remarkable about CRISPR is that it enables you to do gene insertion with a precision that's unprecedented, exquisite precision.
All right.
So let's turn to some other things.
I have the next question, which is, what is your greatest concern with how CRISPR might be used?
Okay. I confess that I thought about this a little bit.